Hello learners, welcome to my YouTube channel Learn Life Science. So this is going to be the third lecture on lambda bacteriophage that I am going to discuss, and the topic is genomic map and lytic as well as the lysogenic switch in lambda bacteriophage life cycle. Okay, in the last two lectures I have already given you a general introduction regarding lambda bacteriophage, lytic and lysogenic switch, as well as the major interaction that appears between the host cell as well as the lambda bacteriophage tail proteins. So genomic map gives you a proper outline or proper localization of various genes that are involved in a defined genetic region. Okay, so the switch between the two states, that is lytic and lysogenic cycle, is mediated by proteins that are encoded by bacteriophage genome. The two regulatory proteins are synthesized by the virus, that is C1, that is acting as a repressor, also called as lambda repressor, as well as the CRO, which is having a regulatory role which controls the repressor synthesis, that is the C1 synthesis. So these two are the major proteins that are involved in the switch, and the thing you have to remember here is that C1 repressor is involved in lysogenic pathway as well as the CRO repressor is involved in lytic pathway. Okay, so these are some of the genes that are present on lambda bacteriophage genome, and it is going to be very easy for you to remember as we will move further. Okay, so the genes are divided according to their time of expression. For example, there is immediate early genes that is going to be firstly expressed, then delayed early genes which is going to be expressed in the presence of immediate early genes, and lastly there will be expression of late genes which is going to be expressed in the presence of delayed early genes. So let's look at one by one. The first immediate early genes involved is N gene. Okay, N gene encodes for a anti-termination protein. By the word it defines that it is anti-termination, means it will allow transcription to go on. Okay, then the second one, that is CRO, that I have already told you, it encodes a CRO protein which is inhibitor of our repressor, that is C1 synthesis. Okay, it will be going to inhibit the synthesis of C1. Okay, then there will be expression of delayed early genes which involved C2, that is also having an important role, which codes for an activator of transcription of C1 and INT. Okay, now here C2 is encoding a protein which is going to act as an activator. Okay, activator of what? Activator of C1, that is our repressor, as well as INT gene. Okay, second is C3. C3 is going to encode for a protein that is involved in stabilization of C2 gene. Okay, that is, it is going to stabilize C2. C2 is going to act as an activator for C1. Okay, and CRO is going to inhibit synthesis of C1, that I have discussed till now. The third one is the INT gene. Okay, INT gene codes for integrase. We all know that integrase is involved in recombination process of different genes. Then there will be cis gene which is involved in encoding a excisase enzyme, which is also having a role in excision and integration. Then there is O and P genes which are involved in DNA replication. Q genes which also acts as an anti-termination protein along with N. Okay, and lastly there will be late gene expression, that is A to J, which is involved in head to tail synthesis. Then S and R genes, that is lysis gene. In this one chart you can clearly see the genes and classify them according to their pathway, that is lytic and lysogenic pathway. For example, N gene is involved in lytic pathway, CRO gene is also involved in lytic pathway, delayed early genes, that is C2 is involved in lysogenic, C3 is involved in lysogenic, and O, P and A to J, S and R involved in Lytic cycle. Okay, so it is going to be really simple for you. Now this is a diagrammatic representation of the genetic map of Lambda bacteriophage. Okay, so what we have discussed about the early genes, it involves N gene and CRO gene. Okay, I have told you CRO encodes a protein that inhibits C1, and then I have discussed about the delayed genes which involved C2 genes, which acts as an activator for C1 as well as the int gene. Then C3 gene is also a delayed gene that is involved in stabilization of C2. Okay, so these are the other genes that is localized apart. The main function or the main regulation is going to appear here. Okay, in the P letter designated here are the promoters. Okay, so before beginning you should know about the operon system. An operon system consists of a repressor gene that encodes for a repressor protein, operator region as well as the promoter region, and there are further your structural genes. Okay, so let's discuss it one by one. So promoter is the region where RNA polymerase binds and initiates your transcription. Okay, sorry, promoter is present right here and the operator is present right here. Let me draw it once again. So promoter is present beyond the operator with close affinity to the structural genes. Okay, and here is the repressor. So promoter is the region where RNA polymerase binds and initiates your transcription and the structural proteins are synthesized. And you might be also knowing that repressor protein is having the ability to bind to operator region and block it in such a way that RNA polymerase cannot move further and there will be no further transcription of structural genes. So here you can see designated P letters are representing these promoter regions for various structural genes. Okay, so here I have mentioned the genes and their roles for you so that you can easily memorize at one look. Okay, in one look. So here lambda repressor is encoded by C1, N is encoding anti-terminator, CRO is the inhibitor of C1 synthesis, C3 is going to stabilize C2. Now I am going to make it more zoomed in to get you understand with the promoters. So as I have already told you, promoter is the region at which RNA polymerase will bind and the transcription will begin. Okay, and the structural genes are going to express their specific proteins. So let's start and focus on these main genes. Okay, now you can see here early genes, that is N gene, here is CRO gene, C1 repressor, C2 activator as well as the C3 stabilizer. Now here is the presence of PL promoter, a promoter that will initiate transcription in left hand side. Here you can see by arrow I have given here, it will start transcription in left hand side. The PR promoter is going to transcribe in right hand side. PRE promoter is present between CRO and C2, that is going to transcribed in left hand side for the transcription of C1. Okay, so the thing you have to remember here is that for C1 expression PRE is going to transcribe. Okay, and then there is also a promoter present PRM, that is repressor maintenance, and as you know with the name repressor maintenance means what? It is going to maintain the level of repressor, that is our C1. So, two promoters are involved in synthesis of the repressor C1. The first one is the PRE promoter and the second one is the PRM promoter. Okay, these two promoter you will have to memorize, and the other one are simple, that is PL promoter, that is going to transcribe the first early genes N. PR promoter, that is also going to transcribe the early gene, that is CRO. Okay. Now, here I have given the full forms as well as the function of various promoters, from which the PL promoter is leftward promoter for early gene N and other leftward early genes. As I have told you, N is going to encode for anti-termination protein, which will allow the transcription of further early genes. The second one is PR promoter, that is rightward promoter for CRO and other rightward early genes. Third one is PRE promoter, that is repressor establishment. Repressor establishment by the name indicates that it is involved in the establishment, means the first synthesis of C1 repressor is going to initiate from PRE promoter. And the important thing that underlines here is the PRE promoter is recognized by RNA polymerase only when there is presence of activator C2. Okay, I have told you that C2 is going to act as an activator for C1, but how it is going to act for C1? It is going to act for C1 by giving PRE promoter its ability to get recognized by RNA polymerase. Okay, means it is an indirect activation of C1 repressor by C2, and also it is going to activate INT, which is I am going to tell you in further. Okay, the second one is PRM promoter, that is a repressor maintenance. It is a promoter that will activate lately and is going to maintain the level of repressor. And the last one is the PI promoter, which is involved in transcription of integrase. Okay, so these are the various promoters that you will have to remember. PL promoter means leftward promoter, PR promoter means rightward promoter, PRE means repressor establishment, which is going to be recognized only when there is presence of C2 activator. Then there is PRM promoter, that is repressor maintenance promoter, and PI promoter for integrase. Now, as I have told you that the immediate early genes are the one that are going to transcribe first. Means what? If I ask you a question that which are the genes or which are the promoters that will be going to firstly recognized by RNA polymerase, then your answer should be PL promoter as well as PR promoter. Okay, because these are the promoters that are going to transcribe the first early genes N and CRO. Okay, so let's get started with the lytic as well as lysogenic switch. Till now we have discussed about the genomic map as well as the various promoters. Now, the first step that is going to happen is the infection of host E. coli cell with lambda bacteriophage. Then the DNA of phage enters the host cell. As I have told you that the lambda bacteriophage consists of various proteins, and I have also mentioned about the protein, that is GPJ protein at the tail fiber, which interacts with LAMB receptor on the host cell. Then the third step the main step that start is that the RNA polymerase is going to bind firstly at PL as well as PR promoter. Okay, and if you remember, PL promoter is responsible for transcription of N, as well as PR promoter is responsible for transcription from CRO genes. Now, gene N codes for anti-termination factor, whose action will allow transcription to proceed in delayed early genes. Okay, then it will cause C3 to be transcribed and as I have told you, C3 is going to encode for a protein that is going to stabilize C2 or protect C2 from action of FTSH. And in the side by pathway you can see SCRO gene causes further transcription of C2, and now C2 will act as an activator for C1 repressor by giving PRE promoter its ability to get recognized by RNA polymerase as well as the activation of transcription from PI. Okay, so let's understand this pathway diagrammatically. So the first thing that will happen is the binding of RNA polymerase to PL promoter and PR promoter. Okay, PL promoter will initiate transcription of N gene. On transcription of N gene, there will be further transcription of C3 gene. Okay, then PR promoter will initiate transcription of CRO gene and then of C2 gene. Okay, and if you remember their function, N is the anti-terminator, C3 is going to stabilize C2, and now C2 is going to act as an activator for C1 promoter by giving PRE its ability to get recognized by RNA polymerase. Means what? This is our first step from PL promoter. This is our second step from PR promoter. The third step will be the activation of PRE promoter and the synthesis of C1. Okay. Now when C1 is synthesized, it is involved in lysogenic pathway, then it will have to inhibit the other genes. Now how it is going to inhibit, let's discuss about it. Now as I have told you, C2 act as an activator which is involved in recognition of PRE by RNA polymerase, as well as it is going to activate transcription from PI. Now the recognition of PRE by RNA polymerase will lead to expression of C1, and C1 is the lambda repressor, the repressor of the genetic regulation system. C1 is responsible for lysogenic cycle. Okay, now C1 is a repressor, means what? It is going to bind to the operator regions and it is going to shut off the transcription of early genes. Okay, so C1 is going to bind to OL, the operator for left hand side genes, and OR, the operator for right hand side genes, and shut off transcription from PL and PR. As I have told you, if the operator region will be blocked, then the RNA polymerase cannot move further. Okay, so this is what here mentioned here is. Now if there is a off transcription at PL and PR region, then there will be no N and no C3, no CRO, then no C2, and if there is absence of C2, there will be no PRE recognition by RNA polymerase, as well as there will be no repressor synthesis. You can see here how C1 repressor is maintaining its own level. Okay, C1 activate the promoter PRM and C1 synthesis continues. So let's understand it diagrammatically again. Here you are going to understand it easily. Okay, so we have discussed until the repressor protein synthesis, which is synthesized by PRE promoter. Okay, when the C1 repressor protein is synthesized, it is going to bind to operator regions. Okay, now the promoter PL is having 3 operator regions, means you can see here there is OL1, OL2, OL3. Here there is OR1, OR2, OR3. The first operator will always be at highest affinity to the structural gene. Okay, always remember the first operator region, that is OL1 and OR1, will be at the highest affinity to the structural gene it transcribes. Okay, then there will be 2 and 3 with low affinity, here also 2 and 3 with low affinity. Now in this section you can see that over OR1 and OR2 imparts the region of PR promoter, as well as the PRM promoter imparts the OR3 operator. Okay, so this was about the diagram. Now the pathway follows that C1 repressor binds with higher affinity to the first operators. Okay, for example, C1 is synthesized initially in the cell, means what, it is now present in low concentration in the cell. Then it is going to bind with high affinity to the first operators, such that there will be no CRO as well as no N synthesis. And then if there is higher concentration of C1 repressor, then it is going to bind to other operator regions, that is OR2, OR3, OL1 as well as OR3. Now remember that the concentration of C1 is increasing in this direction, means what, along with the increase in C1 repressor concentration, their binding with the operator region also increases. This will be at low temperature, this will be at higher temperature. Okay, now if the OR3 region is blocked, what will happen? If the OR3 region is blocked, then PRM promoter, which is also involved in repressor protein synthesis, is also going to get blocked. Okay, so what I am going to tell you here is, first we have discussed that PRE is the promoter which is involved in C1 synthesis, and when the early genes are switched off, the repressor maintenance promoter is involved in its synthesis. But when the concentration is higher and OR3 promoter operator is also blocked, then the function of PRM will also get stopped and there will be inhibition of C1 repressor synthesis. Okay, so this is an important point where C1 is regulating its own level. Okay, so let's memorize it once again, that the first genes that were transcribed were N and CRO. Then other genes involved C3 and C2. In the presence of C2, there was transcription of C1 due to PRE. Okay, there was transcription of C1 from PRE. Now C1 is going to act as a repressor and it is going to block the transcription from other early genes. Okay, then it is going to bind to first operator regions, and along with the increase in its concentration, it is going to block OR3 operator as well as its own synthesis from PRM. Okay, so there was also a question in December 2019 about C1 repressor, that which of the following acts as a positive as well as negative regulator, and the options were about C1 repressor, tryptophan operon, and then LAC repressor, and these were some of the other options, and the correct answer about that was C1 repressor. So this is an important cycle that one should always remember while learning about the lytic as well as the lysogenic switch. Okay, so I have mentioned in a introductory form that C1 repressor blocks synthesis of CRO and also maintain its own concentration at a particular level. The stable lysogen makes sufficient C1 repressor to block not only prophage lytic cycle but also imparting immunity to lysogen against super infection. What mentioned here is that I have told you C1 repressor is involved in lysogenic pathway, means what, it is going to keep the lambda-phage DNA in integrated form with the genomic DNA of the host cell. Okay, and it is also going to impart immunity against super infection, means it is also going to prevent infection of the lysogen with another lambda bacteriophage. When a phage infects a lysogen, excess repressor molecule already present in the cytoplasm of a cell will bind rapidly to the operator site of infecting molecule before polymerase bind to the promoter PL and PR. Now, and I hope you understood this sentence, that now when the concentration of repressor is maximum in the host cell, whenever the foreign viral DNA will inject, the RNA polymerase will be incapable of binding to the promoter region, as there will be already bound repressor to the foreign gene region, and in this way it is going to prevent the super infection. So this was all about the lytic as well as lysogenic switch, that is important point with respect to various competitive exams, and I hope you understood the pathway in a simpler way. If you still have any kind of questions arising in your mind, then you can comment, and if you understood it well, then please like, subscribe and share with your friends. In the next video, I am going to tell you about the retroregulation of INT gene, that I have told you that INT gene is activated by C2, and that is why I am going to discuss the pathway, that how C2 is responsible for expression of INT gene. Okay, so thank you so much, and please stay tuned.